Now, in this module, we will study how enhancer can be located that bind unknown protein. Now, we have studied there are enhancer for a gene which enhance their capability, and they are located at the different positions. Now, the gene-centric strategy is applicable only to enhancer that bind known protein. Now, enhancer, they bind to the protein, and there can be the known proteins and the unknown protein. Now, gene-centric strategy, which is applicable only to the enhancer that have known proteins. And there are many enhancers, definitely, whose protein partners are unknown. So, there are enhancers whose protein partners, they are unknown, and the enhancer where the gene-centric strategy is applicable, where the proteins are known. Now, Pinocchio and colleagues, they found that 167 such enhancer candidates are there which exist for the protein binding. How to test the DNA sequences for enhancer activity? How we can test the DNA sequences for enhancer activity, that is the enhancer which are there. So what is there done that these scientists, they hook them up to LAC-Z and LAC-Z reporter genes under the control of a mouse minimal promoter. So they took the mouse minimal promoter and they hooked them to the LAC-Z reporter genes under control. And then they placed these constructs into a mouse zygote, creating transgenic mice. So they created a transgenic mice using that strategy, and then they allowed the transgenic embryos to grow the embryonic day 11.5. Then strained the whole embryo, which mounts with X-gal to detect the beta-galactosidase activity. They found the strong blue staining with X-gal, and which indicates the abundant beta-galactosidase. Beta-galactosidase, it produced very strong blue staining with the tissues. And they studied 167 enhancer, and they found that of 167 enhancer, only 75%, that is the 45%, they were positive in this transgenic mouse enhancer assay. That is, they produced a strong blue dye of the enhancer, indicating that there is a transgenic activity there. So you can see that the expression patterns which were driven by enhancer, that was discovered by transgenic mouse enhancer assay, how it decreases from one side to another, and then further 47% are there. Now, high success rate of this strategy was that 45% that was achieved by sampling only one stage of embryonic development, because only one stage of embryonic development after 16.5 days, that was 45% was achieved in this one case. Now, this is the good candidate. There are 5,500 more non-coding sequences in the human genome. If we consider it, or if we just consider the human genome, so what we find that there are 550 more non-coding sequences in the human genome that are conserved between human and the pufferfish. Now, there are some genomes, at this point I must tell you, that which are conserved in the species during the evolution. And we found that 5,500 more non-coding sequences, they are in the human genome, and these are conserved between humans and pufferfish species. So this strategy therefore shows great promise for locating enhancer in the human and another genomes, as we have seen earlier, where a transgenic mice, we can get 47%, success or 47% recovery of these genomes. But as it is with the other method, this method has also some drawbacks. And the drawbacks are that it only detects highly conserved sequences. It will detect only highly conserved sequences, the sequences which are highly conserved in the species. And second is, not all important gene control regions, they are conserved. We should also keep in mind that all important gene control regions, they are not conserved. Mainly they are not conserved. And of these, it only detects highly conserved sequences in there. So, in this case, the locating the enhancer that bind unknown protein, there are the different technologies, different strategies which we have studied in this module.